what these plant lectins are. What are the lectins? What are those molecules which have been termed as lectins? And what are the important roles they play in the animal system? It's like there are many therapeutic molecules which are isolated and characterized from the plant system. And the lectins are one of those therapeutic molecule which is being isolated from the plants. So lectins are proteins or glycoproteins that possess at least one non-catalytic domain that binds reversibly to a specific mono or oligosaccharide called a lectin. So the lectins are those molecules. They can be either a glycoprotein or a protein, but they do have a very specific or reversible binding ability to a sugar moiety. So those proteins are called as lectins. So the first lectin was identified in the 19th century from the castor bean by Peter Hermann Stillmark. And in 1954, the term lectin was introduced or coined by Boyd. And the lectin is a Latin word which has the meaning legere, and which means to select. It's like, no, it is highly selective. The property of lectin to have the interaction with a sugar moiety is highly selective. That is why it is being termed as lectin. So if you look into the classification of lectins, based on the carbohydrate binding domain, you have merolectin, hololectin, chimerolectin, and superlectins. If there is a single carbohydrate binding domain, you can say that they are involved in the merolectin category. Hololectin, at least two domains bind structurally to a same sugar. They are known as hololectin. Chimerolectin can bind to more than one domains. And the superlectin binds structurally unrelated sugars. And based on the glycan specificity, you can say that those lectins which bind specifically to glucose, galactose and N-acetyl D-galactosamine, fucose and sialic acid. So with regard to the carbohydrate binding domain and the specificity towards a particular sugar, there are different type of classification for the lectins. So how the lectin is bind to a carbohydrate? On the protein, most of those proteins which are having the carbohydrate moiety, to which the lectin goes and binds. So the lectin is interacting with the carbohydrates. So the interaction site or the combing site of the carbohydrate on that glycoprotein, the lectins will go and interact using, or they do have the hydrogen bonds, hydrophobic interaction and coordinate bonds. So these are the ways by which the lectin is interacting with the carbohydrates on a glycoprotein. So you can see that the structure, if you look into that, the middle of that portion. So this is the place where the lectins are going and interacting. So if you look into the sources of lectin, lectin is ubiquitously present in plants, animals, and microbes. The major source are plants. They are present mainly in seeds and less abundant in other parts. So in most of the cases, we do have seeds and less abundant in other parts. It doesn't mean that we do not have the lectins present in other parts of the plant, because the work which I will be presenting is about the isolation and characterization of a lectin from the leaf. So normally we do have the lectins present in the seeds, but other parts of the plant also you will find the presence of lectins. There are few minor sources in animals, both the vertebrate and invertebrate. In the vertebrates, the soluble integral membrane proteins are lectins. So they are, for example, galactins and selectins. It's like, no, those molecules which form the junction proteins between two different cells. It's like, no, the cells will comes together and attach together to form. For example, we know that how the blood vessel is being formed. So the blood vessel is being formed by the arrangement of multitude of cells connected together using the connection proteins or those junction proteins. So many of those proteins which are involved in connecting the cells together are lectins. And in the invertebrate, mostly they are found in the body fluids and the secretions. And in microbes also, in bacteria, virus and fungi, you will find the presence of lectin. So the lectin are ubiquitously present in almost all the living organisms. If you look into the functions of plant lectins, there are generalized functions and advanced applications. So there are antiviral properties, like there are many lectins which is being identified as antiviral property. So viral coat, that's like, we do have the glycoproteins which makes the viral coat. So what the lectins will do, it will bind to the glycoproteins, prevent the formation of the viral coat. So as a result, the virus will not be able to get into the viral coat, and inhibition of its growth and multiplication will be happening. You have anti-insect properly, it can inhibit the multiplication and activity of the insects. There are anti-tumor, there are many lectins which are being reported to be having the anti-tumor activities. The lectin can go and interact with those. So there are many tumors which are characterized by the presence of certain sugar moieties on them. So there are two ways by which those sugar moieties can be used. One way, the lectin will go and bind to that and induce the cell death process. In the other one, the lectins can be used to identify whether the cell is a cancerous cell or a tumor cell or not, because those lectins or those glycoproteins which are very specifically present on certain tumor types can be detected by these sugar binding lectins. So both for diagnosis and for inducing the cell death, the lectins can be used. It has anti-parasitic activity and antibacterial activity. The same way, there are sugar moieties which are present on the cell membrane. It can interact with that. So once the lectin is interacting with the sugar moieties on the cell membrane, it will lead to the oligomerization. So the oligomerization kind of entrangle the sugar moieties on the cell surface, leading to the pore formation. So once the pore formation has happened on the bacterial cell, the leakage and the bacterial cell death will be happening. So the antibacterial, almost the same way, the anti-parasitic and antifungal activities are also being conducted by the lectins. So antibacterial activity is there. If you look into the advanced applications, so it can be used for the drug delivery. So drug delivery is done because of the specificity of the lectins towards those sugar moieties specifically present on a cancer cell. It is mainly for the treatment of cancers. So you can see that in the cancer cell, you do have the specific sugar moieties. So most of the cancer cells will be producing some very specific glycoproteins or expressing some very specific glycoproteins, and those glycoproteins are present on the cell surface. If you want to deliver a drug, you conjugate the drug along with the lectin. The lectin will have high affinity for those glycoproteins or those sugar moieties present on the cancer cells, so that the drug will be exactly going and sitting on the cancer cell. Cell identification and separation. This is another method. Suppose if you want to separate those cells which are cancerous, or those cells which we need to separate for some experimental purpose. So those cells which are having the expressed carbohydrate, the expressed sugar moieties or expressed glycoproteins on their surface can be separated by a column it's like no in which the lectin is bound. You will be passing the cells through that. The sugar moieties bind to the lectin. It will get separated from the remaining proteins. So the cell identification and separation can be done. There are many techniques like ILAC and LHC. ELA is almost in the way ELISA is being performed. You can have uh, enzyme linked lectinase, that is ELA. So lectin, because of its high specific affinity for a particular carbohydrate, it is possible for us to detect whether the carbohydrate is present on a cell by using an ELISA technique. And you have lectin induced agglutination, lectin histochemistry, or all those things can be done using lectins. You have mitogenic stimulation of lymphocytes. The lymphocytes are involved in the immune reaction, and any molecule which can induce the doubling process or the multiplication process of lymphocytes can be called as a mitogen. Lectins can act as a mitogen for T lymphocytes. They are playing a major role in the defense system or in the immune pathways. You can have blood typing. There are different carbohydrates which are present on the blood. Different type of lectins will be able to differentiate different blood groups. And vaccine production is, it is so far it is not been coming to the reality, but in near future it is possible that some of the plant lectins can be used or can be getting licensed as a vaccine for the treatment of certain diseases. So why these lectins are important tool in cancer research? They are very specific for their interaction with the carbohydrate, and one of the major or one of the significant characteristic feature of the cancer cells are modified carbohydrates. A very specific carbohydrate moiety is present on the cancer cells, which makes them, it's like no, using that sugar moieties, you can detect the cancer cells separated from a normal cell. So the lectins has the ability to differentiate the normal cell from the cancer cell. The specificity of lectin towards these modified carbohydrates on cancer cells can be exploited for designing them as lead molecules in cancer therapy, both in the cancer therapy and in the diagnosis process. Two methods of targeting lectins against cancer cell: direct targeting of the toxic lectin. So there can be the lectins which are toxic to the cells. They can induce cell death pathways, apoptosis, and they can use as a conjugator to target a drug to a particular site for targeting a non-toxic lectin conjugated with a drug. So in that case, the lectin is non-toxic, and the drug what we are conjugating with the lectin will be reaching the target and doing its job. So either the lectin itself can induce apoptosis, or the lectin which is conjugated to the drug will be able to do the treatment or do the therapeutic purpose. So if you look into the cancer applications, the plant lectins in the cancer applications, you can see it can induce apoptosis. Apoptosis is the process in which the internal mechanisms which are present in the cells can lead to the disintegration, the self disintegration, or it is otherwise known as the programmed cell death. The cells will take the decision to die by its own. It is like they are committing suicide. So how the lectins can induce apoptosis is that, as I told you, we need to force the cells to go for apoptosis only those cells which are cancerous. So on the cancerous cells, as I told you before, we do have very specific sugar moieties. So the lectins can go and bind to those sugar moieties, induce the cells to take up the suicide pathway. So apoptosis can be induced by using lectins. Autophagy is another method by which the cells will die by its own. Okay, so in autophagy, you will be having the interaction of more than one cells. They all will come together and uh, die by its own. Apoptosis is normally individual cell based. In autophagy, you have multiple cells involved in that. You have the immunomodulation. That means the immune cells, in the previous slide I told you, it is acting as a mitogen for T lymphocytes. The T lymphocyte replication is very important to maintain the immune system active against the pathogens. So the lectins can induce, or lectin can activate the immune cells. So the immunomodulation. So it will be able to alter the expression. It will be able to change the activity pattern of the immune cell. Though the immunomodulation can be done using the lectins. Lectin array and the glycomase. So lectin array is those lectins which are being used to study the specific carbohydrate moieties present on a certain set of cells. For example, it's like, no, if you look into different cell types, especially the cancerous cell types, it's like, now we know that there are different types of cancers, and in different types of cancers, the sugar moieties, the glycoproteins which are being expressed on the surface of the cancer cells will be different. And the lectin array can be used where we will be keeping different lectins. So each of the lectin will have different affinities for the carbohydrate. So it can be used to study what are the major population of cells which are having the glycoproteins expressed on that. This is very important and this is very useful in identifying what kind of those cells are present in the patient, or what type of cancerous cells are present in the patient. You can have the targeted drug delivery. That is something which we have already told. The specific glycoproteins or the carbohydrate moieties which are present on the cancer cells can be targeted using a lectin which is specific for that carbohydrate, and the drug can be targeted to that. Cancer biomarker. So as we know that the cancer cells are having those sugar moieties very specific to the cancer cells, this can be used as a biomarker. If the lectin is going and interacting with those cell, you can say that the cell is cancerous. So it's a cancer biomarker. So now I'll be talking about some of the experiment which we have performed. So I have used, or we have taken mulberry. Mulberry is a very common plant. We all are familiar with the mulberry. Main use is in the silk industry and one of the commercial source of anthocyanins. It has diverse medicinal properties reported: antioxidant, anti-inflammatory, anti-diabetic, and in the treatment of Alzheimer's, anti-cancer. Mainly the anthocyanins and the flavonoids are having the anti-cancer activity. So we have used mulberry. We have done, so this is the way I'll be completing the presentation. So we have purified the mulberry leaf lectin. So we have taken the leaf, made the crude extract, and followed by the ammonium sulfate precipitation, enrichment of the lectin by IMAC and ida nickel, purification of lectin by SIMDEA. Once the lectin has been purified, it was characterized by hemagglutination assay. So that is one of the major assay for the lectin. All the lectins are capable of making the blood cells agglutinated. So hemagglutination is one of the major characteristics. It is invariably all the lectins has hemagglutination assay. The inhibition of hemagglutination by different carbohydrates. So that will be telling you what is the specific carbohydrate affinity for the lectin. So we have done the hemagglutination inhibition assay for different carbohydrate, and as it is an enzyme, it's a protein, we can check the effect of temperature, pH, and metal ions on the activity of lectin. Lectin is having very high or very wide activity profile. So it is being controlled by temperature, pH, and metal ions. Then we have done the biological analysis, the biological property studies, how the lectin can be, it's like how it affect cell morphology and viability, cytotoxicity, whether it is able to induce the toxic effects on the cell, apoptosis induction, and anoikase induction. So these two process we will see once we go to that. So this is immobilized metal affinity chromatography. So immobilized metal affinity chromatography is nothing but you will have a column, or you will have a system in which you will be using some of the metals which has affinity for histidines which are present on the protein. So the affinity of some of the metals for the amino acid histidine is being used or is being exploited in the case of immobilized metal affinity chromatography, or IMAC. So here we have used, there are mainly four divalent metals: nickel, zinc, copper, and cobalt. So here we have used nickel. The IDA is the ligand, Cepharo 6b is the matrix. So there is a column in which you have a matrix to which the nickel is added through the ligand IDA. You will be taking the extract and passing it through the column. So I told you we have done the extraction, and after that it is being precipitated by ammonium sulfate precipitation. And the ammonium sulfate precipitated fraction, it's 70% saturated ammonium sulfate precipitated fraction was taken. Around 2 milligram of the protein was loaded. And this is how you have got the profile once we check on the SDS page and silver staining. Okay, so we have taken out, we have washed the column, and you can see that this is the marker, and this is the load, and this is the flow, and this is wash, and these are the elutions. 5, 6, 7 are the elution. The 4, 5, 6, 7 are the elutions. Third one is the flow through or wash. So when you do this kind of an experiment, all these fractions, whatever you elute, whatever you think that this is being eluted or this is being purified, your lectin is there, we will be doing the hemagglutination assay to confirm in which of the fraction our protein of interest is present. So what we have taken pH 7 and 6 fractions, which showed hemagglutination activity and used for the next purification step. So among this, pH 6 and 7, that the fourth lane and the fifth lane. So these two fractions were showing, these two elution fractions were showing hemagglutination activity, so that we can assume that the lectin is present in these fractions. So we have taken this pH 7 and 6 fractions separately and taken for the next purification step. The next purification step is the ion exchange chromatography using SIM-DEA. So DEA is a anion exchanger. So this has the affinity for some of the amino acids on the protein. So what we have done, we have taken the pH 7 fraction and the pH 6 fraction. You can see that this is the pH 7 fraction and pH 6 fraction. We had a column. So this is the a very traditional chromatogram. We will be using it. We will be doing it using a chromatography equipment, and we pass it through the column, wash it, and elute it. So when we eluted it, you can see that there is a very okay. Okay, so you will be eluting it out, and you can see that the elution, this is the second one is the load, one is the marker, second one is the load, 3 is the peak. It's like, no, this is the peak 1, and this is the fourth one is the peak 2. So you have a peak 2 and a peak 1. So where you can see that there's a single band. In both the cases, you can see that a single band is being present in peak 2. So there is very some changes in the arrow. You should see that this is the peak 1 and this is the peak 2. Okay, the arrow is somehow it is being changed. And here you can see that this is the peak 1 and this is the peak 2. So you will be getting the single band. You will be getting a single band in the case of elution fractions in both the pH 7 fraction and the pH 6 fraction when it was passed through SIM-DEA. So this is the purification table. So once you do this kind of biochemical purification studies, we will try to calculate how much is the activity. So it all start with the crude extract, and in the crude extract we have this much total protein, and the total activity, it's like, no, this is irrespective of all the hemagglutination activity. So when you have this much protein, you have very high hemagglutination activity. The specific activity is the total activity divided by the quantity of the protein. That is the specific activity. And the yield, always the first crude extract will have the 100% yield, that is we will consider it as 100% yield, and it is not purified. So the purification fold also will be considered as 1. So now we have the 70% ammonium sulfate precipitation. Once it comes to 70% ammonium sulfate precipitation, we have 318 milligram of protein and this much activity, specific activity, and the percentage yield. Because you can see that from 405, it has reduced to 318. So the percentage yield has reduced from 100 to 79. So in the same way, IMAC pH 7 and SIM-DEA, only the activity increases, the specific activity is getting increased, the total activity is getting decreased, but the specific activity is very important. And the percentage yield, the activity percentage yield is also getting reduced, and the purification fold is getting increased. So this is how you will represent when you go for a protein purification in the normal purification procedure. You will take into consideration of all these different factors and make a purification table. So this is the purification table for one of the fraction, and this is the purification table for the another fraction. Okay, so this is pH 6 and the previous one was pH 7. So there is nothing, all these values will be same from here onward. The IMAC pH 6 onwards, the quantity will be different. Same GAE also, the quantity will be different. But the specific activity, you can see that it is getting increased. And the purification fold is also getting increased. So now we had two proteins from pH 7 and pH 6. So it is necessary for us to identify whether these proteins are same or not. So what we have done a 2D page analysis in which we have loaded pH 7 alone, we have loaded pH 6 alone, we mixed pH 6 and pH 7. So if there is a change in the structure or in the size or in the charge, you will be having two protein dots which will be present on the 2D page when we are analyzing it. We could not see any separate protein dot on any of the gel, which means on all the gel, the protein dot were present on the same place. So it has been concluded that both the pH 7 and pH 6 fraction, we are getting the same protein. Somehow in one of the, it's like when we have done the IMAC, the proteins are being, or the fractions are being eluted in two different fractions in pH 6 and pH 7, but both are the same protein. So we have then confirmed the nature of the protein by doing a reduced and non-reduced gel. The reduced and non-reduced gel will be helping you to tell whether it is a monomer or it's like a monomer or a multimer. So here we are seeing in both the cases, in the reduced condition and non-reduced condition, we are seeing only one band. That means the protein, the lectin, what we have identified and isolated is a monomer. Okay, so this lectin is a monomer. And this is the hemagglutination activity where we have taken the blood groups, means the blood sample from different blood groups, around 2% RBC suspension is being used. And we can see that we will be diluting it gradually. And at a titer of 2 to the power 6, we got agglutination of A positive blood. 2 to the power 4, we got a B positive, and 2 to the power 6 again, O positive blood, we got the hemagglutination happening. Now we have done the hemagglutination inhibition as using different carbohydrates. So this is like, so how the hemagglutination is happening when we are adding lectin to the RBC suspension is the carbohydrates which are present on the RBC membrane are being bound with the lectin, which lead to the agglutination of RBCs. Okay, so on the surface of RBCs, we have sugar, which is helping or which is binding with the lectin, making the precipitate or making the hemagglutination. Now what we have done, we have taken different sugars, incubated with the lectin. After that, those incubated sample was used for the hemagglutination assay. So wherever it's like galactose, galactosamine, and N-acetyl D-galactosamine, when we have used 25 millimolar concentration, it was not able to do the inhibition. Okay. Then we have analyzed, it need to be done. It's like the carbohydrate analysis also to be done. So there is a staining called the PAS staining. It is periodic acid shift staining, which will be staining the glycoproteins. So there is a positive PAS staining. You can see that there is a 43 kilodalton protein, and which is ovalbumin, and purified MLL. You can see that a little bit in the higher side. It's around 58 kilodalton. You can see those two proteins found to be glycoproteins. And there is a phenol sulfuric acid method by which you will be able to use sugar or glucose as a standard to find out what is the sugar concentration on the cells, or sorry, on the electric. So we have studied the effect of temperature on the hemagglutination activity of MLL. So we can see that at up to 40 degree, 20 degree, 30 degree and 40 degree temperatures, there was no effect or there is no reduction in the activity. But at higher temperature, 50, 60 and 70 degrees Celsius, the activity has come down to 50%. And at 80 degrees Celsius, it has come down to around 20-25%. So it is sensitive to high temperature. Effect of pH at 6 pH, it's like no basic pH, it was facilitating the activity of MLL, that is the lectin was active at higher pH, but it was having very less activity at acidic pH ranges. Effect of different metal ions. So we have seen that aluminum, ferrous, 2 plus and Fe3 plus. These three chemicals are promoting the activity of MLL, but all the other metal which we have tried were inhibitory, or it was not giving any upgraded activity for the MLL. So we have later studied the effect of MLL on different cell culture systems. What is the effect of MLL? So in this, you can see the first one is the morphology. We have studied the morphology. We have used two different cell lines, MCF7 and SCT15. You can see the middle, it is plus MLL and plus cisplatin. Cisplatin is the positive control. It's like no, cisplatin is a chemical which is known to have inducing apoptosis on the cells. It is a medicine which is being used in the chemotherapy treatments. So cisplatin in the laboratory is normally being used as a positive control. We do have MLL. When we have treated MLL and cisplatin on MCF7 cells, you can see that. So this is a control where the cells are normally grown. In the case of HCT15 is also there. But when the MLL is being added to the cells were detached, the pseudopodia has been gone, and the cells were coming into the floating stage. And the same result is there for the cisplatin also. And we have found out that the GI50 value of MCF7 is around 8.5 microgram per ml. When you have used MLL at a concentration of 8.5 microgram in MCF7, the cells were showing this kind of response. And in the case of HCT15, it was 16 microgram per ml. So those two are different cell types. Then the DNA fragmentation. So once this cell is showing this kind of a morphology, that means the cells are going for the cell death pathway. So the DNA fragmentation assay was done using acridine orange. Okay, and we can see that we have MLL. This is the lectin added cells, and this is the control, and this is cisplatin added cells. The DNA get, so you can see that in the normal cells, it will be round structure, and if the DNA is getting fragmented, you can see that the dots are there. Very small dots will be present, and you can see that a lot of dots are there in some of the cells in both the case of cisplatin treated cells and MLL treated cells. We have tried the membrane integrity also. So the membrane integrity is, there is something called phosphatidylserine. It's a molecule which is present inside the cell membrane, which get exposed once the cells started going to the apoptotic pathway. So these phosphatidylserine can be identified by using annexin 5. So the annexin 5 will be identifying the phosphatidylserine, which is coupled with a fluorophore. So the MLL treated cells and the cisplatin treated cells, you can see that the phosphatidylserine is being identified using the fluorophore combined annexin 5. The membrane integrity is being lost, and this is also the translocation of phosphatidylserine annexin 5. You will be able to see there is a merging of the cells. You will be able to see that in the merged cells. So this is the annexin 5 treated cells, and this is carboxyfluorescein treated cells. When you merge the cells, wherever the cells are having the translocation of phosphatidylserine, the cells will be showing a different color. And in this case, the control, you can see that it is green in color. Then we have seen the apoptosis, and now there is a process called anoikis. So anoicus is a process in which it is again a type of programmed cell death induced upon cell detachment from the extracellular matrix. Okay, the cell will be detached from the extracellular matrix. So this is actually the way by which the cells are moving from one place to another, a process called metastasis. So metastasis is very significant in the case where you will be getting the tumor cells moving from one place to another. So the metastatic process is like, no, the cell will be getting detached from the tumor. It will be moving to a new place, getting attached to a new place and multiplying there, making a new tumor there. So anoicus is a process which is again another type of programmed cell death in which the cell will be detached from its original position and undergo apoptosis. The cells will be died by itself. It acts as a defense mechanism by preventing detached cells readhesion to new matrices. So when the cell is undergoing anoicus, the cell will not be able to get attached to a new matrix and the metastasis will not be happening. So in the case of MLL, when we have treated these cells with MLL, we have seen that the cells are getting detached and undergoing apoptosis. So this is again the MLL induced apoptosis in MCF7 cells. So there is OD 560, it is MTT assay in which we will be taking the cells, treating with MLL and doing the MTT assay. Once the cells are there, it will get colored because of the MTT. So you can see that those cells which were treated with MLL 5 microgram and MLL 10 microgram, the cells get detached so that when the plate is washed, the cells were gone away. So that the color made by the addition of MTT was very less in the case of MLL treated cells. So it shows that control, you will have very good purple color formed by MTT, and MLL treated cells, the cells were detached and lost. So it shows that it can induce anoicus. So this is again the time and concentration dependent cell detachment, the anoicus, which is we were talking before. So this you can see the control. The cells are very highly compact. There is no space between this. When you have 2 microgram, 5 microgram, and 10 microgram, you can see that how the cells are getting detached. One by the time it's like, no, this is 4 hour, 8 hour, 12 hour, 24 hour, and 48 hours. So by the time 48 hours, you can see that the control cells are very compact, and the cells which are treated with the 2 microgram, 5 microgram, and 10 microgram, you can have the cells getting detached and lost. So the cells are not present. So you can see that in 4 hours, all the cells are there. This is the same plate which have photographed after 4 hours of treatment, 8 hours, 12 hours, 24 hours, and 48 hours. And this is the same graphical representation of the same thing. You can see that in 24 hours, the control is same. The control is there, and the other types, MLL 2 microgram, 5 microgram, and 10 microgram, very less number of cells present in that. And in 48 hours, you can see that again, the control is same, and the treated cells are very less. Detached cells takes the intrinsic route of apoptosis. So intrinsic apoptosis, I told you again, this is the process by which the cells decide to go for suicide. So there are different methods by which we will be able to check into that. So the intrinsic route of apoptosis is the apoptosis starts from inside. There is a lot of stress molecules which can be produced inside the cell. There are a lot of reactive oxygen species, and whenever it's like, no, when the cells are getting detached, it can induce a stress which will be activating some of those pro-apoptotic molecules in losing the membrane integrity of mitochondria, release of cytokine C, and leading to the activation of caspases. So we can see that when you have MLL 5 microgram, 10 microgram in the mitochondria, the BAX, BAD, and HSP60, all these proteins are pro-apoptotic proteins which are expressed in the mitochondria. In the cytosol, they are not present also. Okay, so there are different methods by which we have identified it. The DNA fragmentation is happening. The DAPI staining is there. The agarose gel electrophoresis is there, where we have taken out the DNA and analyzed it. We have acridine orange and ethylene bromide staining, which shows the integrity of the membrane. Annexin 5 staining, the phosphatidylserine, Western blot analysis for different molecules. The caspase non-activity. Caspase molecules are those molecules which are involved in the cleavage of proteins. So they are actually molecules which are doing the apoptosis. Okay. So the Bax activity, caspase activity, all these things were upregulated when we treat the cells with a mulberry leaf lectin, which shows that the cells are taking an intrinsic route of apoptosis for or as the effect of MLL treatment. So coming to the summary, we can say that a lectin of around 56 kilodalton from mulberry leaves was purified using chromatography, both IMAC and SIMGA, with a yield of around 61.46%. The sugar inhibition study showed that MLL is specific towards galactose, galactosamine, and N-acetyl D-galactosamine, and retains its H activity over a wide range of pH and temperature. The hemagglutination activity, we have seen that it was present over a wide range of pH and temperature. The growth inhibition by MLL in MCF7 and HCT15 cells, which was at a range of 8.5 microgram and 16 microgram. The growth inhibitory activity of MLL on MCF7 and HCT15 is due to the programmed cell death. So the MLL could induce programmed cell death or apoptosis on MCF7 and HCT15 cells. MLL further shown to be inducing anoikis in MCF7 cells. So I told you anoikis is a process by which the cells will get detached and goes to apoptotic pathway. So when the cells were treated with MCF7, treated with the MLL, the cells were detached and lead to the apoptotic pathway, leading to the cell death. So I would like to acknowledge Dr. Deepa, who has done most of the work, Suresh Kumar, who was involved in the purification, Dr. Priya, she was working along with me for the cell culture studies, Dr. Priya Sudhagaran for some of the assays, Dr. Sandhya for some of the assays at IIC Bangalore, and DBT for the funding. Thank you so much, and I'll be happy to take the questions if there is some.
Yeah, thank you so much, sir, for the highly informative and interesting session. Dear participants, now the session is open for discussion. You can put up your questions in the chat box, or you can directly ask the questions to the speaker. Please come forward.
I think the topic is a little bit away from the plant science only. So, but it is important that the students should have some idea about the interdisciplinary research areas. So this is the first part is with the plant, and the second part is with the animal system.
So dear participants, you can put up your questions in the chat box. You have time again. Yes, sir, that's a question.
It's mulberry leaf lectin. It is not MML. It is MLL. It is the mulberry leaf lectin. It is easy to convey the things with the short form. So it is MLL. It is mulberry leaf lectin.
I'm Dr. Robin Jose from Kerala, and I wish to ask one question.
Yeah, sure.
So you mentioned that mulberry has some anti-diabetic property.
Yes.
So can you mention the compound which is responsible for this anti-diabetic property? So how we have to administer the leaf as medicine?
The compound wise, I may not be able to give you the idea, because normally the studies which were conducted, they have used the extract. It's like, no, they will be taking different extracts and using it with the mammalian model systems, something like diabetes induced mice models, and seeing that how these extracts are changing the induced diabetes, and means how it is helping the mouse model system to survive with the induced diabetes. Exactly, I'm not sure about which is the molecule responsible for that. Maybe I'll be able to look into that, because it's like, no, diabetes is not my area. So I'll be able to look into that, and I definitely, if I get something, I will pass it on to Sunilal.
Thank you, sir.
Yeah, there is a question. Is there any difficulties in isolating specific lectin using chromatography? So the isolation of lectin is, there are different methods. See, what we have used is a method in which there is an affinity. See, we do have the amino acid histidine in all the proteins. So we do have histidines in all the proteins. So the histidine is an amino acid which has affinity for the metal ions. So the affinity for metal ions of histidine can be adjusted, can be manipulated by using different buffer systems. So what we does, we use a certain buffering conditions in which we facilitate the interaction of histidines on lectins to the metal. Here we have used the nickel. To nickel, the histidine will bind so that the remaining proteins will be going down. So that is how we have purified it. But the best way to purify a lectin is, once you identify what is the specific glucose moiety to which this lectin is binding, you can make a column with the glucose or with the sugar moiety. Pass the lectin through the, or pass the extract through the column. To that sugar moiety, the lectin will bind, and it can be purified. So this is a reversible binding. You can use different pH buffers to dissociate the interaction between lectin and the sugar. So the most easiest method to purify lectin is the sugar affinity method. And can we use lectin for all types of cancer therapies? See, it depends on, because it's like, no, the lectins are plant derived molecules, plant based molecules. So what is the issue when you use a plant derived molecule or a plant based molecule for the therapeutic purpose? So that is why it's like, no, the lectin studies have been conducted, or it is being conducted for many years now, but so far the FDA or any of those regulatory agencies have not permitted the use of any lectin for the therapeutic purpose. Only in the diagnostic purpose it is being used. The problem is, the lectin, being a plant origin molecule, may have some negative impact or negative effects on the mammalian system. It can interact with so many other different moieties. So what the scientists are trying to do is that they will be manipulating the structure of the lectin in such a way that only the carbohydrate binding domain will be retained, and the other domains, it's like, it's a glycoprotein. It could be possible that it's a big protein. So when you infuse a big protein as a therapeutic molecule to a mammalian system, it can adversely affect the mammalian system. So just because of that, it is not been in practice the therapeutic purposes with the lectin. Lectin maybe in future, that's what I'm saying. The affinity coding domain alone can be used for getting it bound to the sugar moiety. So it could be possible that in future it is engineered lectin could be used for the treatment. So far no lectins are being allowed to go for the treatment.
Okay, thank you so much, sir.
Thank you. Thank you.
Now, I kindly invite Dr. Deeptamal MJ, Assistant Professor from the Department of Botany, to propose a vote of thanks.
Good morning, everyone. I'm extremely delighted to express our deep sense of gratitude to our dear sir. this section on plant lectins in disease biology. It is an interesting section. So explain lectin used as biomarker to detect cancer cells. What are the applications in lectins? Different techniques are adopted for different disease identification, purification method by immobilized metal affinity chromatography, page analysis, and extent chromatography. The section created the realities that plant phytology and biochemistry are the field of the present and future that will replace microelectronics and many fields with tremendous applications potentials. Through this conference, I wish everyone to enhance the innovations of science in our daily life and existence. On behalf of the management, principal, Department of Botany, and all Fatima family, I extend our heartfelt thanks to you, sir. Thank you very much.
Thank you, ma'am.
Thank you, sir.
Thank you, Dr. Deeptimol. So now we wind up this session. We move on to the next session by 11:30 a.m. The link will be opened 15 minutes prior to the session, and the feedback link will be provided during the second session. So thank you. Thank you. Thank you very much.
Thank you, ma'am.
With the advent of new molecular regulations of plant secondary metabolites, a world of possibilities have opened up for the diagnosis and treatment of many diseases. This session looks forward at the field of nuclear medicines and clinical applications in research, thus to enhance the science of physiology and biochemistry to widen the scope of plant sciences. We feel proud to have such a vibrant, energetic, and eminent speaker, Dr. Padmanabhan Mohanan, scientist, Seoul National University, South Korea. I invite Dr. Nirmala Jayarani, Associate Professor, Head of the Department of Botany, Fatima Mother National College, to introduce the guest of the day. Ma'am, please.
Hello, can you hear?
Yes, ma'am.
Yes. Okay. Good morning, everyone. Good morning, Dr. Padmanabhan.
Good morning, ma'am. Good morning, everyone.
So I am really happy to introduce Dr. Padmanabhan to the gathering. He has done his PhD Biotechnology from the School of Biotechnology, University of Gyeonggi, Republic of Korea, and also he has done his advanced diploma in molecular diagnostics from the Department of Biotechnology, Bharathasan University, and he has had his MSc Biotechnology from St. Joseph's College, Tiruchirappalli, and currently or at present he is the postdoctoral researcher at the Plant Genomics and Breeding Institute, Seoul University, Seoul, Republic of Korea. He also had teaching experience at Trichy St. Joseph's College, and he was also a career advisor and a best researcher in genomics, and he had very good publications, and he is also very much interested in the area of plant genomics. And really I had a chance to attend one of his talk while he was delivering in Tamil Nadu, and I was very much impressed by his talk, and I contacted him, and at once he accepted my invitation, and he said, surely I will one day deliver or present his findings. So really we are very happy to have you, Dr. Padmanabhan, and I am also really happy because he is my junior, saying that I also did my MPhil in Trichy St. Joseph's School, and he has also done his MSc and MPhil from Trichy St. Joseph's College. So we are college mates actually, though I was so much senior to him. He is an young researcher, very much in developing in the field of genomics. So on behalf of all the faculty present in the Department of Botany, the IQC of Fatima Mother National College, and the principal and the management, and all those participants present here, I welcome Dr. Padmanabhan for his interesting and informative talk. Padmanabhan, please.
Thank you, Super Senior and Dr. Nirmala, and thank you, Dr. Sajuma, for introducing me. This really a pleasure, honor for me to, join Fatima College presentation again, one more time to India. Without further delay, let me begin my presentation. So I hope you can see my screen.
Yes.
Right. Right. Very good morning or good afternoon to everyone, wherever you are. And I am Padmanabhan Mohanan. So I'm currently in Seoul National University, Seoul, like Professor Dr. Nirmala introduced to me. And thank you for this opportunity. And I'm very much honored to present in Fatima's College as well. Right. So I chose topic like molecular regulation of secondary metabolite biosynthesis. Due to the pandemic period, like the current pandemic period, actually give a very good, very good opening of like knowing about the secondary metabolites or beneficial compounds present in the plants. And in India, we have a lot of plenty of medicine. It's not a lot, plenty of medicinal products, medicinal plants, and we have medicinal compounds as well, like the Kabasara Kudineer or whatever the prescriptions been suggested to COVID-19. So they have all the mixture of like plenty of medicinal compounds and its extract, which benefits actually to the human being, and it's very well effective against all the viral or bacterial pathogen or whatever it is. Right. So we are studying about all these like antioxidant properties or anti-cancer, anti-diabetic, whatever the properties what we are studying are still at the primitive level, like very basic level we are doing it. And actually, Dr. Ahmed, I cannot remove my mask because I'm in the lecture hall. So I'm sorry, I could not remove my mask. Maybe at the end of my presentation, just I will remove it a little bit. Right. So with the adventure of many research, what we are doing in India, about this finding out these metabolites or activities, but we are lacking a majority about finding out the effective components or compounds present in the plants and how we can improve or increase the production of those plants in all these medicinal plants. So I chose this topic so that I can give an idea or overall view of like how we can approach a particular compound, particular process, how we can do it. So that's my motto of this presentation. And finally, I will briefly introduce what I am doing here and what I'm working with plants. I'm working with a plant, Panax ginseng. So everybody knows about ginseng it's in revitalized and so on. Right. So let's get into the topic. So as you can see, the plants are actually just a moment. Right. I call like plants are superstars because they are autotrophs. They can do their own photosynthesis. They can protect themselves against all the biotic and abiotic stresses in the classical mechanism, like how they perceive the sunlight, and then they convert that into carbon source in the leaf, and they releases oxygen for us to breathe. And though during this process of plant respiration, so we are getting glucose and the products which plants produce for its own purpose, we are also using it for our own beneficial activities. And apart from these primary metabolites, they also have the secondary metabolites, which is predominantly majority of molecules what the plants have produced, because they cannot move away from invading pathogens or any other stresses. So they produce a secondary metabolite with the help of hormones. So how these primary metabolites governs their growth and development, and in such a way, hormones also regulating their own primary metabolite productions as well as secondary metabolites. And primary metabolites in turn supports the production of secondary metabolites, and secondary metabolites protects the plant. So secondary metabolite interact with the environmental conditions so that they can produce against the defense against the stress compound. So this is the basic biology of what is the plant has, primary metabolite and secondary metabolite. Right. So what actually these plants contain? What are the secondary metabolites the plants contain? There are plenty of diverse molecules what plants have. Basically, they've been characterized into like a few compounds such as phenolics, alkaloids, saponins, tritepenoids, and flavonoids, glycosides, etc., etc. So among the phenolics, most of these compounds also have the beneficial role, but some of them or few of these compounds as well as have their negative aspects in the human life as well, because it's a secondary metabolite. It's a different molecule for the plants. Obviously, they produce to attack or fight against the herbivores or predators what the plants encounters. So basically they have all the beneficial activities which we are requiring, the humans, in normal life. So secondary metabolites are rich source of pharmacologically active compounds. That's what we have been seeing, and that's what I was telling in the previous slide as well. Right. So let's give not if not detailed look, just give a brief look about all these like a metabolite element, and then we move on to the regulation portions. Then plant phenolics, there are almost 8,000 different plant phenolics known so far, and among the plant phenolics, they've been divided into flavonoids and non-flavonoids compounds. So flavonoid compounds, as you can visibly see in plants like anthocyanins or flavonols or flavonols, basically color compounds. And non-flavonoid compounds are phenolic acids, lignans, and tannins and lignins, which you cannot see it, but you can taste it. So like a bitter color, bitter taste. So these are their structures. Most of them are organic molecules. So structurally, they are diverse. Structural variation is existing in each subgroup, and based on the structure, they've been given a different name, like isoflavonoids or flavonols or lignans or tannins. Right. So when we come back to the beneficial aspects of plant phenolics, so the quercetin, well-known compound, which we see in the tea, coffee, and everything. So those are very much beneficial, and they are abundant in onion, broccoli, and the apple as well. And catechin and cyanidin glycosides, like these compounds, anthocyanin, and everything. And those compounds are really beneficial to human in terms of like fighting against adipogenesis or angiogenesis or autophagy or even the cancer, or even in the skin treatments. So these are the beneficial parts of this plant phenolic compounds. If you want to know more about these beneficial effects of these phenolic compounds or plant phenolics, each section or each metabolite also has their own detailed reviews. So viewers are advised to refer those. And then when we talk about alkaloids are famously known as anti-cancer molecules in terms of vinblastine, vincristine, and codenine or quinine, all those compounds. So they are diversely present and across the species, including Fabaceae, Asteraceae, Solanaceae, Apocynaceae, and Luteaceae, which we are normally use in our regular life. So they are like pretty much useful for humans as well. So apart from that, the very important and most abundant secondary metabolite, terpenoids. Terpenoids also present in the plant. It's present eventually in all the species of plants, all the genus of the plant, even in some fungus species. Some bacteria can able to produce terpenoids as well. Because the core pathway leads to produce terpenoids is a common central pathway which links structural production of cell wall, which includes the basic primary metabolite and comes below glycolysis. So this is pretty much present in every living species. So terpenoids are dimers and polymers of 5-carbon precursors. They often evaporate from plants. You can smell it, or you can see it through latex or gum. And they are present in the carrot or all the color compounds, celery, and everything. And the ginsenosides, one of the active pharmacological compounds of ginseng, is also a class of terpenoids. They are called triterpenoids because of the 3 rings, triterpenoids. And abscisic acid is also very much known for plant stress. They are also terpenoids as well. So yeah, they use it out in hot sunny days, so that it can sometimes turn as toxic to pathogens. Right. So what are the benefits again? These are the beneficial components like myrisine, epenin. Even though they are all called with a different name, they have a common activity in terms of humans, like anti-inflammatory, antidepressant, and anti-tumor activity. And they are sometimes used as a pain relief compound as well. So those are the beneficial terpenoids. Okay, let's get into the main topic. What we are going to see, like how these terpenoid or how these secondary metabolites been regulated or produced in the plant. Usually, all these secondary metabolite production is tightly controlled in plants because it's an energy consuming process, first of all. And second, when they are overproduced in the plants, that might lead to deleterious effects to the plants as well, because sometimes they can also damage the plant cells, and that's why they are stored in the vacuoles predominantly, or in the outer cell wall, or even in the epidermis layers of the plant, or sometimes in the roots, so that plants aerial parts, like such as the leaf, stem, or fruits or flowers, whatever they are produced, they cannot be affected by these secondary metabolites. So if you are talking about tightly controlled process, that definitely involves transcription factors, because transcription factors is like a ping pong mechanism. So once it attached to some molecule, or once it's attached to some other bind dimer, then it goes to do like DNA binding activity, and then they promote the transcription of particular genes. So here is a list of R2, R3 MIP protein. MIP protein and BHS proteins are the well-known transcription factor family, which involves in most of the production of secondary metabolites such as lignins, coumarins, or phenylpropanoids, and anthocyanins and glucosinolates, and so on. So all these R2R MIP3 are present from model plant Arabidopsis, which have the homologs in all the plants as well, all the medicinal plants, which I will explain it in detail. So glucosinolates is one of the famous compound from Brassicaceae family. Arabidopsis also belongs to Brassicaceae. In secondary wall production, there is another MIP transcription factor, phenylpropanoids, secondary cell wall, and planktocyanins, anthocyanins, flavonols, and all these secondary metabolite production is been tightly regulated by R2, R3 MIP type transcription factors. And you can see they are also belongs to subgroup. Actually, based on the phylogenetic tree, they classify them into subgroups, and they sometimes have the BHLS domain, sometimes not, and sometimes have WD40 repeat domains, sometimes they don't have. So R2, R3 MIP, BHLS, R2, R3 MIP, WD40 domains. So those transcription factors are involved in production of secondary metabolites. Right. So what are the necessary or essential amino acids or domains which are required for this production of secondary metabolites being listed up here? And as I said, some of these core components are conserved, evolutionary conserved throughout the plant species in between secondary different secondary metabolism. So subgroup 2 consensus sequences these are the amino acid sequences which you have to look for when you find an R2, R3 MIP transcription factor. And if you find those R2, R3 MIP transcription factor containing this particular subgroup or sequences, then you can name them to particular, or you can label them to particular secondary metabolic pathway. Then you can start to work on the activities or the effect of these transcription factors. So that is the notification from this slide. So all these subgroups have all these domains. So mostly they are all basic amino acids. And apart from that, there are two groups of BHLS domain. One is activator and another is repressor. The same transcription factor which binds to the promoter of any metabolite gene, metabolite biosynthetic gene, they can either activate or they can repress the activity of the gene. So activator type BHLS domains has classically these conserved amino acid sequences across species: Arabidopsis in Siamase, Arabidopsis and Bison sativum, Arabidopsis and rubber like Hevia brasiliensis, Arabidopsis and Solanum species in Piper species, and etc. And this is the repressor type R2R MIP3 transcription factors. They also have conserved domains. So even though the protein has abundant amino acid sequences, amino acid profile within the particular domain of a protein, I'm sure you also know, or you all know about the terms of these domains and motifs. Within the particular domain, there are several motifs which, based on that, you can recognize them as either it can be activator type or repressor type. Even a single amino acid changes, which might lead to the activity of the total protein, protein structure of the activity of this protein, and that might determine the role of this particular transcription factor or proteins. For example, I'm sure some of you know about the flowering mechanism in plants. So one of the key genes of flowering mechanism is called flowering locus T gene. So they have a domain motif sequences. So the flowering locus T domain. So among those domain, there is a particular amino acids. When it is turned from one amino acid, the same activator FT can turn into repressor FT, which blocks the production of flowers in plants. So that's how the protein chemistry works. That's how the protein structures work. Eventually, even though you see as an amino acid, is the structure binding which interacts with the particular protein or domain that might leads to production of the effect what we desire for. So that's why these authors listed out the repressor domain and activator domain, and in order with the sequences where they regulated in diverse metabolic process. Right. So what is the take-home message from here? So when you find any secondary metabolite biosynthetic genes which might be regulated by R2 or MIP3, first to find out the domain for R2 or MIP3 sequences, and among the R2 or MIP domain, there is a motif where you can find either it is an activator or repressor, and find those things, activators or repressor R2 or MIP3. If it is activator, then works towards the activity of this transcription factor binding and transcription of the particular gene, or if it is repressor, then find out the promoter where it is binding and see the expression of the gene if it is blocked, or how this mechanism is working. Or if it is a repressor, then it might interact with another co-binding protein as well, so that they both action on repressing the gene expression. So let's see some of the examples of how this secondary metabolic control and in the plants works. So there are two classical examples, or the recent papers which I have read. So one include AP2 ERF transcription factors. These transcription factors are pretty much known for the developmental regulation of the plants. Like they actually, when they express during the development stages of the plants, and they helps for the growth of the plant. And how this also involves in binding or regulating the cluster of secondary metabolism that's been listed out based on the research in tomato and potato plants. Right, API transcription factor families are three types, three subgroups. There are AP2 domain, AP2 ERF domain, and there is a linker, and again AP2 ERF domain, and only one domain that is called DREB or DREF or ERF domain. And raw proteins, they have another basic amino acids domain and the AP2 ERF domain. And among these AP2 ERF family, they've been known to bind to these sequences in the promoter that is called cis-acting element. So APT binding genes, binding domain is this one, and GCC box in DRECRT box, CE1 box, and RAV box. So the GCC box, GE box, or G box sequences are very much popular among transcription factor family, which can bind to those those sequences in the promoter. Right. So among the cis-actor element, drought responsive element that is called DREB, in Arabidopsis sequences, they've been maybe you can see here. Yeah. So Arabidopsis sequences been known in Arabidopsis, CMIs, Orisa, and wheat, Triton sativum, everything is known. And low temperature responsive elements and ethylene responsive elements, so on. So as you can see here, there is no information about how these how these cis-active elements involved in secondary metabolism, but you can see most of them are involved in the development and regulation of the plants, like drought responses and low temperature response and ethylene responses and hormone responses in consensus side responses in plants. In this paper talks about AP2 transcription factor as an activator of triterpenoid, like terpenoid indole alkaloid biosynthesis in Cantharanthus roseus, which we are getting vinblastine, vincristine from this plant, Cantharanthus roseus. So how they find these sequences is that, so this is the common pathway which I talk about, like they come from the glycolysis, this terpenoid pathway. So membranoid pathway in pyruvate 3P, and they produces MEP, and then goes for further process and leads to production of vinblastine and vincristine. So here, in particular region where it can promote the vinblastine action is that they bind it to ZCT domain in the TG box, like ARCA transcription factor. And when this ARCA transcription factor is phosphorylated by this AP2 family domain, and they promote the transcription of this particular gene, and in turn leads to production of vinblastine and vincristine. So here is the transcription factor where they act on in the total pathway. So they are all jasmanic acid elucidated mechanism, elucidated pathway. So when you induce jasmanic acid in the plants, they convert the downstream signaling being transduced by MYC2 transcription factor, and MYC2 again bind to ORCA family, that is AP2 transcription factor. They present as a cluster, so which I will explain in the next slide, like coming slides. So they present as a cluster. So once activated, and they keep on induces activity of other transcription factor, which in turn produces these responses, these enzymes which are involved in production of streptocidin, tryptamine and anthralamine and logonic acid, cyclolagonine, which are the precursors for the end product in bloodstream immunity. Right. So how they find this AP2ERF transcription factor, which has a role in inducing the TIA, that is tryptanoid alkaloid pathway, is that so they try to map all the AP2ERF families from Arabidopsis, Cantharanthus, tomato, and potato tobacco. Their target plant is Cantharanthus, and then they find out some of them are linked, some of them are related to each other, and when they screen it completely in the Cantharanthus, and they find these clusters, these particular clusters, which are pretty much related, and they falls under AP2ERF family in GAME9. This is very interesting transcription factor, which I will explain in the next part of this research. So GAME9. So this GAME9 loci is present in tomato and potato as well, because they are all Solanum family, they are sisters. So they produces some of the secondary alkaloid like steroid glycoside molecules. So there's those came, and those ERF family are present in the tobacco, is produced in, which is involved in the production of nicotine metabolite, that is nicotine locus. So again, I said like those ARCA family or AP2ERF transcription factor families are present as clusters in tomato, in potato, also in tobacco, which they produces nicotine locus. And then they find out these ARCAs are present here. Then they started to characterize all these ARCA genes and want to find how this is related, right? So ARCA3, ARCA4, ARCA5, these dots represents the production in the metabolite enzymes. So ARCA3, when they are induced after the jasminic acid induction, so they promote the production of tryptophan, tryptamine, and then stricotylcysteine, so like that. And when they find out the target compounds, admelicin in cantharanthine and taberosine, you can see that jasminic acid induction is enhancing the production of these particular compounds, but not cantharanthine, right? So then they find out what is the reason behind or how this is being regulated. Again, I said as a transcription factor, they will bind to the promoter sequences. So they try to find out the important promoter sequences such as STR promoter, PMT promoter, and QPT promoter, where they are binding in the sense. STR is where? Okay, let's go back. Here, STR promoter in PMT. PMT is PMT in tobacco, is here somewhere here, and then QPT promoter. So then they find out that this ARF221 in tobacco, also ARCA3 from Cantharanthus roseus, they are all similarly binding to this particular promoter family, and they have also the G-box domain sequences. And these promoters, they activate the production of these particular metabolites, what they are looking for. So this is one example how they find out. And then when they look at the DC-rich motif, where the necessary binding of this ARCA3 takes place in the JRE motif, then they find out ARCA3 itself cannot bind, and they bind together with ARCA5 as well. And later they find out that this ARCA3 and ARCA5 works together. Like once activated ARCA3, they activate ARCA5, and then together they go on bind to the STR promoter and the cantharanthine promoter to enhance the production of secondary metabolic process. So this is how they promoted. So they mutated this ARCA3 domain, derepression domain, and then find out if they can activate or not. See, together with ARCA5 and CRMYC2A, which is the upstream of jasminic acid signaling here, which they can bind to the activity of this promoter, LAMP promoter, and they enhance the production of cantharanthine. This is how they find in final mechanism. So they find the initial transcription factor ARCA3. In ARCA3 also enhances during jasminic acid production. In cantharanthine and the other steroid glycosides are also increased during the jasminic acid productions. And so they find out the relation how this jasminic acid production is transduced to ARCA3, and ARCA3 is inducing the production of these metabolites. They find out that CRMYC2A, that is Cantharanthus roseus MYC2, in ARCA3 binds together, and then they enhances the activity of the secondary metabolites. So that is the one part. So they're checking overexpression analysis as well. Okay, so they bind to the group 9 ERF, that is ARCA. They bind to P box and CS1 box and GCC rich box in tobacco as well as in Cantharanthus roseus. So this is a example where these promoter regions are bind in the plant. But let's move on to this game9, which I said is a sterile alkaloid binding transcription factors, and how this is increasing the isoprenoids production in melanin pathway. So this is the sterile alkaloids present in the plants, like potato. So when you expose to the light, these green color produces in the potato, that is glycoside alkaloids, so which become toxic to humans. So when you find a potato which is green in color, don't buy it or don't eat it, because they have rich in glycoside alkaloids. So that alkaloid which is toxic to humans. So when it is in the dark condition, they will not produce. So when you buy the potato, just keep it away from the sunlight. So there are many 50 different like the sterile alkaloids have been identified in potato, both in wild and commercial variety, and tomato also 100. So both tomato and potato are sister families. So these authors, what they find out that they try to map this loci where this is game transcription factor is located in between tomato and potato. And they find out that the arrangement of the transcription factor in the chromosomes of tomato and potato are very much related, and they are enriched in particular region of these game transcription factors. So in tomato it is called game, in potato it is called sphingolacta, like a steroid glycoside transcription factor family. So when they overexpress this particular game4, which is involved in production of this precursor, which leads to produce tomatidine, which is green in color when tomato, and when it is ripe, it becomes red. It is degraded and produced in red color. So they increases the production of tomatidine. And then the same group, let's find out, try to find out the same loci which is responsible for producing green color sterile alkaloids, the sterile alkaloid which produces green color in potato, and they find out there is a relation of called game9, that is game9 transcription factor family. And when they match tomato and potato transcription factor, and they find one particular loci, like such as SL game9 and potato game9, which is very much related or interacts with most of the like a transcription factor family, and then they start to characterize this particular transcription factor. So they publish another paper from the same group in Nature. So game9 regulates the biosynthesis of steroid alkaloids and upstream isoprenoids in the plant. So it's again an AP2ER of transcription factor family. So they have like this many genes co-expressed together, in the sense they express in the RNA-seq data is very much related when they are upregulated or downregulated. If it is upregulated, all these genes are upregulated. If it is downregulated, all these genes are downregulated. This is tomato loci and this is potato loci. And they involved in production of cycloethanol synthase. Is another steroid glycoside which is involved in producing steroid in the plant. The expression profile shows that they are highly expressed in the leaves and buds, which is bud is like the potato buds, which you can see in the potato plant. So buds and leaves in the peel as well, and when it's in the flesh. Right. So this is the metabolic network which involves in producing like this is the terpenoid family until the sesquiterpenoids, and then from sesquiterpenoids all this compound moves to cytosol to produce steroids. In among the steroid pathway, there is a intermediate compound. Actually, it's not an intermediate compound; it's a precursor compound, cycloethanol, and then which leads to produce cholesterol, and camposterol, which molecule, which forms the basic plasma structures of the plant. So when they overexpress this particular domain, what is the level of cycloethanol? They find out that cycloethanol is not much highly overexpressed, but the beta amarin is produced. Beta amarin is here. Beta amarin rings are here. Beta amarin is overexpressed in the overexpression line, and camposterol also highly expressed in particular overexpression lines. And when they do it in the tomato profiling line, when in the RNAi, they knock out the game9 transcription factor, the production of cycloethanol, camposterols are all increased. So the function is directly opposite to each other. So how? In transgenic potato, when they overexpress this game9 transcription factor, which is present in both potato and tomato, they overexpress in the potato, the camposterol level is higher and cycloethanol level is down, which leads to down regulation of this cycloethanol, but cholesterol is highly expressed. How? Because when this game9 is overexpressed, and they also overexpresses other like steroid glycoside interacting transcription factors such as game11, game6, game4, game12, and game 2, which produces the alpha-solanine and alpha-chaconine, which is a toxic compound for steroid glycosides in potato. They're also highly expressed. And in the transgenic tomato, the same mechanism happens. HMG are also expressed, which is a top upstream of terpenoid metabolites, and followed by the beta amarin synthase, and then tomatidine level as well. So in potato, like cyclooctanon synthase are over highly expressed, and which leads to production of solanine metabolite. And in tomato, the metabolic genes were upregulated, and upstream MEV pathway genes and game gene clusters are also upregulated when they overexpress when they overexpress these particular game metabolite. So when they compare the co-expression data from like a potato and tomato, they find two important genes. There is like cellulose synthase-like domain and SSR2, and here GS80 domain. When they match the localization in the chromosome 7 of tomato, they find that the cellulose synthase like a gene, which is very much aligned towards or aligned with the game transcription factor family. So among the many genes, 27 genes were common between RNAi and OXM lines in tomato, and those are all steroid metabolism associated genes. And cellulose synthase like genes, which is here, and SSR2, steroid side chain reductase, and saturase, desaturase, which leads to production of cholesterol, are all very important genes which are co-expressed together in tomato and potato. Among 27 genes, there is one E3 ubiquitin protein ligase family, which controls the HMGR enzyme, are also found. And that is why when they overexpress these lines, these upstream genes were transcribed highly, also along with game factor family. And because of the cellulose synthase family, like genes, which are together aligned with the structure of the game transcription factor family, they increase the production of other steroids as well. So when they compare the SL, like the tomato MIG2 and tomato like potato game transcription factor family, they increases the production of steroid glycosides, and might be some other transcription factor also involved. And apart from that, they are also activates the production of their own transcription factor family and upstream metabolite, which produces the precursors to produce cyclooctanoids and terpenoids. And that's how this potato and tomato the potato and tomato steroid glycosides are accumulated. Right. So take home message from here is that we have seen two examples of AP2 ERF transcription factor family, which either interacts with another MIG2 domain, MIG2 transcription factor family, which induces expression of particular secondary metabolite genes, and also in turn they also regulate their own production, their own subfamily of genes, because they all produced in the clusters of sequences, clusters of arrangements in chromosomes. So that's how they increases the expression patterns of all different transcription factor family, which leads to enhances the expression of steroid glycosides in plants. Okay, finally, this is the final presentation. So finally, the work what I'm doing in SNU, the Seoul National University, related with the ginsenoside biosynthesis. So ginseng is it belongs to Araliaceae family, the same family, which is the same order where the carrot and like a celery or coriander leaves and everything is present, which is also similar in terms of their architecture of the leaves and flower. As you can see here. So that belongs to the genus Panax, and the name is ginseng. So it is a like a perennial plant. So it lives under the soil for like many years until you dig out. So they produces important metabolites, ginsenosides, which is a triterpenoid metabolite, which I have like gave a short note in the previous slides, and gintonin and glycopyrrole. They are abundant. They are like actively researched for their activity. So they have prolonged life cycles, and after 3 years they start to flower, and then they increases the root weight, and they eventually produce these roots. The ginseng, what you see in the revital, is not actually Panax ginseng. That is another related genus to the ginseng, and that is also called Siberian ginseng. So that is a tree. In that ginseng and this ginseng is completely different, and activities are totally different between each other. So Panax family has many other many other species. So most predominantly, what we know is what are the families which are used for medicine as well as commercial purposes are these 5. Panax ginseng, which is in Korea, Panax pilpifolis, which is in America. Actually, not in America, America-Canada border. It looks like that, which is very, very similar to in the structures of the root. In Panax noto ginseng, which is in China, Japonica in Japan, and Vietnam, which is in Vietnam. So the present lab, what I'm working with, they sequenced the whole genome of Panax ginseng, and they find out that Panax noto ginseng and Panax vietnamensis, which are two-end, it's a diploid, they might have evolved together to, or mixed together to produce Panax ginseng, which is a tetraploid. These two plants are tetraploids, foreign in nature. However, there is a considerable differences between the genome of noto ginseng, but we're very much close to vietnamensis. So there must be an intermediate plant which is lost in the evolution. So that is the story. The detailed structure or detailed metabolic pathways, how these ginsenocytes are produced in the ginseng, is been very well characterized. If not the genes, but most of the metabolites are pretty much mapped. The classical MEA pathway, terpenoid genes, which terpenoid metabolite pathway, which leads until squalene and squalene epoxide, like 2,3 oxidosqualene, and from there onward there is a branch point which is called damarine synthesis, which produces damarine ring, which closes the ring of oxidosqualene, and that leads to production of diverse ginsenoside metabolites. This in Korean ginseng. If this DDS is not present, then they will produce all the sterols, which we are usually seen every plant, which we have also seen in the potato and tomato plant. So this damarine synthesis is a time-consuming process which converts the damarine in ginseng. So where it is produced, where it is located in ginseng means it is mostly stored in the vacuoles and also outer epidermis in the roots. So they looks like that when they are fully matured, around 6 years old. So these are the active components. In medicinally important active metabolites are RG3, CK, RB2, RH1, and RE. These names are named after the presence of the glucose moieties in the basic tritepenoid structures, which you can see in the many reviews. What are the activities? There are plenty of activities for these ginsenoside molecules. Each ginsenosides are having their own specific activity, signaling mechanisms, and everything. And each structures, each form of ginseng also have their own unique activity. The white ginseng is a fresh one, red ginseng is the boiled and dried one, and black ginseng is boiled and dried in hot air oven. So it become a blackened structure. So what is the purpose of doing this is that they reduces the complex structure of ginsenoside to the simpler compounds such as RG3, CK, or RG1, which can be easily absorbed by human body rather than excreting in the via the urine. It can be readily absorbed by the human body, and it leads to beneficial activity. However, red ginseng is the predominant one, and it's been produced in the extract and tablets which everyone consumes in Korea. So these are the activities in cancer, diabetes, and neurodegeneration, and so on. So each ginsenoside metabolite, they have different activity, and that activities depend on the glucose structure, which binds here, 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 here. So everywhere, based on the glucose moieties binding locations, their activity and binding to the signaling molecule is different, and that leads to activities different. So there are many genes being listed out which can induces the production of ginsenosides in ginseng, and this is the life cycle and how the ginsenosides are accumulated in the ginseng plant. So right from the seed, they also have ginsenoside RE. This is a highly active pharmacological molecule as well, and good for skin as well. So they produces most of the ginsenoside within 3 years, and then once the year goes, they store it in the root rather than in the leaves, because root lives for longer time, not the leaf. And these are the genes which are characterized in simplified form, pathway mechanism I have given here. Right. So they've been characterizing how these ginsenosides are produced and what are the age and how during the age they are produced. So you can see year 1, year 4, year 6, all are accumulated differently in the leaves as well as in the roots. So to simplify everything, this produces ginsenosides in the beginning of the first year, and then stored in the root at the end of 4 year or 6 years. This is the proportion. Right. So my role is that to find out what are the transcription factors, or how these ginsenoside genes are activated in activated or actively transcribed in ginseng. So previous publications by my senior found out that when you overexpress the jasmonic acid genes, they induces the expression of these particular genes which are responsible for producing ginsenoside. And she found that some of the transcription factors might involve. So my role after I come to Seoul National University is that so we screen many RNA sequence libraries, and then we found out that there are certain transcription factors which are co-expressed, which I have given in the previous studies as well, co-expressed together with the important genes involved in ginsenoside biosynthesis. So one such gene is this BHLH domain, which I'm currently working on and still work in progress. So it has a BHLH domain motif in the 3' end, which is binding or co-expressed together with squalene epoxide, which produces 2,3-oxidosqualene, and dominant diol synthase, which produces dominant diol, that is a precursor for ginsenoside, and so on. So this is a mixed type BHLS domain. So when we check their expression pattern, there are 2 paralogs. As I said, it's a tetraploid. So probably you may assume that it may produce 4 genes, 4 active genes of expression. Sometimes 4 or sometimes 2 only express. So in case of BHLS transcription factor, there are 2 domains, 2 genes which are actively expressed, BHLS1 and 2, and they express predominantly in the 1-year and 5-year-old peak. They are actively abundant. And in terms of stress as well, cold stress is increases in the expression pattern of these genes, also in the heat. So we assume that they may have a role in enhancing the ginsenoside biosynthesis in ginseng. So I screened their developmental aspects because, as I have shown in the previous slide, the 1-year-old and 5-year-old ginseng has accumulated or produced ginseng at the different parts of the ginseng. One is in the leaf, 1-year-old, in 6-year-old in the root. So during the developmental stages, from winter to spring and to summer, where the active photosynthesis happens, how the ginsenoside genes expression pattern and this transcription factor expression pattern is related. So I found that this BHLH1 and 2 is constitutively expressed. However, they are abundant in the summertime, during the summertime to like spring to summertime. That is the ratio, which is correlated to the expression pattern of all these ginsenoside biosynthetic genes, Ginger1, SS1, C3, and TDS1. And this is another way of inducing the ginsenoside biosynthesis in ginseng. So the adventitious roots of ginseng, when you treat methyl jasmonate, is the elucidation mechanism, which is very much common to all the most of the secondary metabolite production. When you induce methyl jasmonic acid, they induces the expression pattern of these terpenoid genes. As you can see, the expression is started at 12 hours, and they start to accumulate until the 48 hours. So which correlated with the expression pattern of this BHLH1 and 2, which is constantly expressed. However, BHLH2 is predominantly accumulate during the time course of time, which is related to HMGR1. So I characterize that the missing transcription factor could be BHLH1, which might bind to these 3 genes. That might lead to accumulation of ginsenosides in ginseng. So that is what I'm working on. Now I'm working on how this BHLS is binding to these promoter regions of these genes and what is the role behind it. Right. With this, I conclude my presentation. So this is my lab in SNE. Thank you for listening. So if you have any questions or anything to ask, you can ask me now.
Thank you, sir, for such an informative session. The presentation on molecular regulations of plant secondary metabolites embarked a new dimension in the field of molecular biology and phytochemistry. Dear participants, now the session is open for discussion. You can put up
Yeah,
the-
Dr. Ahmad have asked some questions in
yeah,
the
I have
chat
two
box.
queries. I have
Yes,
asked
Dr. Yeah.
two queries. One is regarding the genes which are present for secondary metabolites. Are they housekeeping genes or inducible genes?
No, they are not housekeeping genes. Mostly secondary metabolites cannot be housekeeping genes because once it overexpressed or overproduced in the plant, that might lead to deleterious to the plant. So those are inducible genes. So that's what the aim of this presentation is that. So is that they are inducible, and some of the transcription factors like BHLS, MEAT2 or ERF transcription factor or APT domain, they might induce they could induce the production of like these metabolites in the plant. So they are not housekeeping genes, they are inducible genes.
My second query is what is the amount of product, means in micromoles or millimoles, if you are performing some histochemical test, because primarily if we want to detect the secondary metabolite in any part of the plant, we need to undergo the histochemical test. So for the histochemical test, what is the quantity or what amount of a secondary metabolite is expected?
Probably in micrograms, microgram per gram dry weight or milligram per kilogram dry weight, something like that. So usually histochemical studies are being done for these color compounds, for example, anthocyanins or flavonoids. But usually these secondary metabolites, you can do HPLC or UPLC or GCMS or LCMS to find out the amount of metabolites present in the plant. So in that case, microgram per gram. Yeah. And if you talk about micromole, that is the activity of these secondary metabolites towards particular conversion reactions. For example, converting squalene into oxidosqualene, how much micromole needed, something like that. So those are two different entities.
Particularly in case of your studies, when you have reported genes, alkaloids, those alkaloids, are there any fixed amounts?
No, actually, steroid alkaloids is not my study. I taken from Israel group. Okay, ginsenoside is my study. In terms when I talk about ginsenoside, I express them in the microgram per gram because they are accumulated in the roots or leaves. So I do HPLC usually to detect them. So those are microgram per gram dry weight. Yes.
Thank you so much. Thank you.
Yes, doctor. Thank you.
Any questions? Okay, thank you, sir. Now I
Thank you, ma'am.
kindly invite.
Thank you, ma'am. Please unmute.
Okay, thank you, sir. I kindly invite Dr. Ruben Jose, Assistant Professor, Department of Botany, Fatima Mada National College, to propose the vote of thanks.
Good afternoon, participants. We are listening to Dr. Padmanabhan, and his deliverance is on molecular regulation of secondary metabolite biosynthesis. He is working as a postdoctoral researcher in Plant Genomics and Breeding Research Institute, Seoul National University, Seoul of South Korea. We are hearing an interesting deliverance from a young, enthusiastic researcher. He explained about He explained the significance of phenolics, alkaloids, terpenoids, etc. So he emphasized on the regulation of secondary metabolite biosynthesis, different steroid alkaloid biosynthesis, especially regulation of SGA content by GAIM9 gene. Finally, he concluded with his work on ginsenoids and ginsenoid biosynthesis. We on behalf of Fatima Mada National College, Fatima Mother National College Department of Botany, we extend our sincere thanks to Dr. Padmanabhan for his enthusiastic deliverance. Thank you, sir.
Thank you, Dr. Raman. Thank you. Honored.
Thank you very much, Padmanabhan.
ഇതുവരെയും
Thank you,
മാസ്ക് മാറ്റിയില്ല.
Thank you, ma'am. Yeah.
Thank you, sir. Thank you, everyone.
അതാണ് ചിരിക്കുന്നത്. ഒരാളെ
Dear
ജസ്റ്റ് ഒന്ന്
participants,
ഐഡന്റിഫൈ ചെയ്യണം. അത് ഒരു ഇതാണ്.
now the
Okay.
feedback link is open in the chat box.
So I
We
will
have-
leave now.
Humble submission to the organizers is it was the conference on plant physiology. It is my area of interest. So why can't it the recordings can be put into the YouTube, and you can we can use it for our classroom teaching also, because some topics are like not some topics, whatever the 4 lectures were there, all the 4 lectures were very excellent, and actually we can use in some time course of time for classroom teaching also. So if it could be possible, it is a wonderful meeting. You can record it. The recordings can be posted on YouTube and sent to the participants. It's my humble submission, if it is possible. Thank you.
Okay, sir. We'll do it. Now the oral presentation is going on, and we have a concluding session at 4 p.m. today, and all the participants can join the concluding session in the Google platform itself. The same link can be used. So we can, this time to wind up the session. So thank you so much. Thank you all the speakers, especially Dr. Padmanabhan, and all the participants. And especially thank all the faculties of Department of Botany, Fatima Mada National College, for the hard work and the time that they have taken to conduct this virtual seminar, virtual conference successfully. So thank you so much. Thank you, everyone.
Thank you, ma'am. Thank you for the opportunity. Let's meet one more time.